Bueno, buenos días a todos. Bienvenidos a este nuevo evento del capítulo regional de la Acoustical Society, Spanish Speaking Acousticians in Americas. Hoy vamos a presentar a la doctora Julianna Simon, que es parte del Graduate Program in Acoustics de Penn State. Vamos a hablar un poquito de nuestro capítulo regional. Nos establecimos en 2019. Tenemos más de 200 miembros en este momento. Y tenemos miembros en todas las áreas técnicas, que son 14 según cómo divide la Acoustical Society sus áreas técnicas, y predominantemente tenemos miembros en el área de acústica arquitectónica, Engineering Acoustics y control de ruidos. Ahí lo que ven son nuestras redes sociales, el Facebook, nuestro canal de YouTube, donde van a poder ver las grabaciones de estas presentaciones, y nuestra página web, donde van a poder encontrar más información sobre nosotros. Para hacerse miembro de este capítulo, no hay que ser miembro de la Acoustical Society, ni tenemos que pagar ningún costo de membresía, ¿sí? O sea, es totalmente gratis. Entonces, simplemente tienen que entrar a nuestro site, a ssasa.org, y hay un botón que dice Únete, llenan sus datos, y con eso ya son miembros de nuestro capítulo y van a recibir nuestro mailing list con todas las novedades. Nosotros siempre estamos buscando nuevos miembros que quieran ser parte de la junta directiva. A final de año, este año vamos a tener un cambio, una renovación de autoridades. Así que si están interesados en participar en nuestro capítulo, nos pueden escribir al mail que está en pantalla, es un Gmail, y pueden vincularse con nuestro capítulo. Bueno, este webinar va a ser presentado en inglés. La doctora Simon va a hablar en inglés, pero tenemos traducción simultánea al español. Lo que hay que hacer es conectarse con el símbolo del globo terráqueo en el Zoom y apretar el canal al cual quieren escuchar. Si es en inglés, van a escuchar a la doctora Simon hablando en inglés, y si apretan español, van a escuchar la traducción. Si quieren hacer una pregunta, pueden ponerlo en el Q&A. Yo lo voy a estar monitoreando, y también el chat. Pero en general las preguntas van a ser respuestas una vez que termine la presentación de la doctora Simon. Así que ahora voy a cambiar, un segundito, voy a cambiar al inglés. Share screen. Bueno, so we're presenting today Dr. Julianna Simon from the Graduate Program in Acoustics at Penn State. I'll read her bio pretty quickly, but Dr. Simon received her Bachelor of Science degree in Bioengineering from the Washington State University in 2009, and her PhD in Bioengineering from the University of Washington in 2013. Her thesis was entitled The Thresholds and Mechanisms of Tissue Injury by Focused Ultrasound, including her discovery of the mechanism of tissue fractionation in boiling histotripsy. After finishing her PhD, Dr. Simon received a National Space Biomedical Research Institute First Award Postdoctoral Fellowship to investigate kidney stone detection with ultrasound in spaceflight analogs. In 2017, Dr. Simon joined the Pennsylvania State University as an assistant professor in the Graduate Program in Acoustics and Biomedical Engineering. The goal of her lab, which is called the Biomedical Acoustics Simon Lab, or BASiL for short, is to apply the fundamentals of acoustics to develop novel ultrasound diagnostics and therapeutics. Some of her current projects include understanding the role of bubble nuclei in pathological biomineralizations, developing ultrasound to treat tendinopathies, diagnosing and treating heterotopic ossification, and activating and imaging phase change contrast agents. Dr. Simon has authored more than 25 peer-reviewed publications and more than 130 conference papers in national and international conferences. So the topic of the talk that Dr. Simon will give us today is called Diagnostics and Therapeutics Ultrasound Technologies for Earth and Space. So I leave Dr. Simon to share her screen. and talk us about her research.
Excellent. Thank you for the introduction, and thank you for the honor of speaking to you today. As Fernando mentioned, I will be talking about diagnostic and therapeutic ultrasounds, technology for Earth and space. Some of this work is work that I did at the University of Washington that I've taken here, and then I'm collaborating, of course, with groups at Penn State to try to bring in their expertise and make the technologies actually move into the clinic. So without further ado, if you're not too familiar with diagnostic ultrasound, the first recorded use was in 1942 by Karl Dussek in Austria, who looked at transmission through the skull to diagnose brain tumors. So kind of a surprising first instance of ultrasound use, because we know that through the skull is very absorptive and attenuative, but he was trying to use ultrasound to diagnose brain tumors, and that's this image over here on the right. In 1976, so over 30 years later, is when the US started to set safety limits by the Food and Drug Administration. And we continue to improve and revise these safety limits as we understand where is injury caused by ultrasound. So ultrasound continues to improve. Nowadays we have 4D imaging, so space and time, and they continue to improve contrast and resolution, so you can get images that are very clear, like this picture of a fetus on the bottom right-hand corner. Therapeutic ultrasound is less commonly known. It was actually discovered before diagnostic ultrasound, where Langevin found that fish were dying during sonar development. And so that was the first recorded case of ultrasound kind of as a therapeutic, where they found that fish died when the sonar was put into the ocean. 1938, still before the case of imaging with ultrasound, Zeiss started to find some bioeffects in the eye with sound. And nowadays, therapeutic ultrasound is used clinically worldwide. In the US in particular, it is approved for uterine fibroids, bone tumors, essential tremor, and some other brain diseases are getting more and more use in the clinic. How does it work? Well, basically it focuses the energy, the acoustic energy, like a magnifying glass, so that you get very hot spots or very high energy acoustic waves at the focus area without damaging the intervening tissue. And so this is the big advantage over therapeutic ultrasound over other modalities, is we can treat deep in the body and we elicit a therapeutic response through hyperthermia or heat, or through bubble activity, where the bubbles oscillate and collapse and can literally be used to chew up the tissue. So that leads to my motivation. I'm very interested in spaceflight, and many of the biological changes occur in spaceflight also occur on Earth. And so ultrasound is the only diagnostic and therapeutic that can fly. It's so prevalent and available in developing countries because it is portable, unlike so many of our diagnostic and therapeutic technologies. So my main goal while I'm here at Penn State as a professor is to try to develop these ultrasound diagnostic and therapeutic technologies, and not just in the lab and not just understand the fundamentals, but also be able to translate them into the clinic, into space, into developing countries, etc., so that it's widely available for people to use. So I'm going to split my talk up into two main areas. One is looking at controlled soft tissue fractionation, called histotripsy, where basically I'll talk through some of the theories and experiments in bulk tissue and talk about ongoing work in my lab. But then I'll also switch gears and talk more on the imaging side about bubble-based ultrasound imaging, specifically looking at the Twinkling artifact for some different areas. So high intensity focused ultrasound, also known as HIFU, is generally the word used when we're talking about using that focused energy to thermally denature or coagulate tissue. Basically, we're cooking the tissue similar to how you could cook dinner, but we're cooking a very isolated region, killing the cells, killing the proteins, and basically not allowing the tissue to be able to survive. The other side, as you know, is mechanical fractionation. So this is where we get those bubbles oscillate and collapse and can literally create holes in the tissue without any sort of thermal border. Now, there's two different ways of mechanical fractionation. One was developed at University of Michigan, and that's called cavitation cloud histotripsy. They really look at using the negative pressure to try to expand and collapse these cavitation bubbles. The other approach that's somewhat synonymous, but slightly different, is called boiling histotripsy, where we're using the heating of the bubble, so high absorption from nonlinear heating to create these bubbles, which then causes more bubbles to form, and the interaction with those bubbles literally chews up the tissue, just like cavitation cloud histotripsy. So how does this work? Well, if you take a spherically curved transducer or an array and focus them into the body, nonlinear propagation effects cause these waves to become very shocked. This is just one waveform measured. We can see the peak pressures around 60 megapascals positive, minus 18 megapascals negative. So these very high pressures that happen only in that very small focal region in the body. With boiling histotripsy, we're relying on that peak positive. And because it's a nonlinear wave, shock wave heating goes by amplitude cubed. So it depends on the linearity of the tissue, the frequency, but the heating rate does depend on the cube of the shock amplitude. Because of this, we can actually reach or exceed 100 degrees C at that very small focal spot in a matter of milliseconds. And once we reach 100 degrees C, we get boiling. And so here we see that a boiling bubble has formed once we reach that temperature. Now, here's what this looks like in a gel phantom, where we can image it with a high-speed photography. So we get this boiling bubble. That's a very large bubble that forms. But then in front of it, we get all of this cavitation activity, because once we have the bubble, it acts like a pressure release interface. The positive becomes negative, and we get significant cavitation activity. These work together synergistically to chew up the tissue and create a hole. And so the boiling bubble actually ends up in what's known as this tail of this tadpole shape. Cavitation activity is what chews up this head region. And we get this very small hole in the liver. Now, the size of this hole we can change by changing the frequency of the transducer. If we do histology, where we cut thin slices of the tissue, stain them so we can look at cells, we see there's a very sharp boundary. Even though we used heat, we get a very sharp boundary between the normal tissue and the homogenized tissue, where we don't see any tissue structures remaining. The question I worked on extensively in my PhD thesis was to understand, how does a relatively large boiling bubble, millimeter diameter, fractionate tissue into its submicron components? And the hypothesis that was raised was this idea of atomization. So ultrasound humidifiers use atomization, where these small droplets pinch off, and that's what causes these droplets to be out into the air to increase the humidity. Once we add focusing of the ultrasound wave, if we try to do this in water, so this is a focused ultrasound transducer in water aligned at the tissue or the water air interface. If we image that, we see the radiation force creates this actual fountain. But then if you look off to the side, you see all of these small droplets pinched off in what's known as atomization. So bringing this all together, the proposed mechanism I looked at was this idea that this HIFU that would focus a wave, and that focus wave would heat through absorption the tissue at the focal area. As this wave continued, the tissue would reach or exceed 100 degrees C, after which case this heated region would change into a vapor cavity, have a phase change. And once we have the vapor cavity, we're still sending the ultrasound wave, and the ultrasound wave then would have that radiation force and create this micro fountain. And that, along with the cavitation activity, causes these droplets to pinch off in what's known as atomization. Now, if you hit this same spot over and over again, you continually are recirculating these fountain projectiles and getting it to literally chew up the tissue. And so tried this experimentally. So I drilled a hole through tissue, hit from underneath with a focused ultrasound transducer. And what we found was kind of surprising, that the tissues really looking like this liquid type behavior, we're getting it so that we get this fountain, we get this atomization. As soon as HIFU ends, because this is an artificial hole drilled through tissue, we see that the hole remains. But basically this repeated circulation of focusing it, having this fountain form, having these droplets pinch off, is what's causing the tissue to be chewed up. and it does result in tissue erosion. So if I just change that circular hole to a flat interface, I see that as I increase the number of pulses, the size of the hole, this divot I'm creating in the tissue, increases, suggesting that yes, we are eroding tissue by this phenomenon. Now, one question that remained was whether that actually worked out into the liquid slurry, and it does. So if we collect these fountain projectiles, we may have some cells remaining initially, but if we recirculate them, eventually we can chew up the tissue into its submicron components. Now, we have validated this in a pig model. This was an open abdomen, so we weren't trying to go through the fat and skin initially. We took an ultrasound transducer, we had imaging probe for alignment, focused this ultrasound energy into the tissue. Now, one of the advantages I didn't mention of histotripsy over a thermal mechanism is that we can monitor the bubbles with the ultrasound imaging. So we get this hyperechoic spot that's showing that we do have bubble activity. We have that change in impedance, that's why it shows up as bright. And we can use that to monitor the treatment where we're seeing this hyperechoic, these bubbles remaining after each pulse. What we found in the porcine model, when you first finish the treatment, you see this kind of bruised looking area, but you can remove, use a gauze and blot out that tissue structure, and it actually, you see remove the liquefied contents, and you see that yes, we did chew up the tissue, if you will, but it did spare some of the connective components. So our main outcomes trying in vivo versus the ex vivo tissue is that thermal effects were less pronounced in vivo. This wasn't really a surprise because we do have blood flow that's pulling heat away as we're trying to heat, but also that connective tissue structures were spared. And so the liver is not just cells, but cells held together in clumps by extracellular matrix. These extracellular matrix clumps are remaining behind. So we don't get a perfect whole, but we do get this whole separated by these septa from the connective tissue structures. So what had worked so far, what's completed so far in boiling histotripsy is that we do have an understanding of the mechanism. We can control in soft bulk organs exactly the area that we're treating and can get an effect. We can control it to the extent that if you're bored in the lab one night, you can write your name in the tissue. We can monitor it with B-mode imaging because we are using bubbles. And we've tested in an in vivo porcine model. Ongoing work is to test it in renal and hepatocell carcinomas, going after cancer type mechanisms, because we can use this to destroy the primary tumors non-invasively. Now, where my lab has taken over a lot of this work is that the same tissue structures that are spared in vivo also might be a limitation for some applications. So one of the first areas we went after, which was funded through the National Institutes of Health, was this idea of rotator cuff tendon injuries. So rotator cuff injuries are the third most frequent pathology treated by physical therapists, with an annual treatment cost of over $7 billion in the US. The big challenge is that tendons never really fully recover. If you have a tendon or ligament injury, it will never be restored to its pre-injured levels as far as the loads it can support. Now, if you have a tendon injury, current treatments are something called dry needling, where they take a very thin needle and repeatedly pepper the tendon, trying to break up scar tissue and allow healing factors to come in and infiltrate. Another one is extracorporeal shockwave therapy, where similar to ultrasound, they're sending in this nonlinear acoustic wave into the body. Now, there's no imaging alignment. They just kind of align based on eye of sight or where it hurts. And of course, the most extreme is some sort of surgery, whether it's a minimally invasive surgery like arthroscopy, where they put in probes, or even open shoulder type surgery. The challenges with all of the current treatments, though, are one, they're generally invasive, and success rates are very mixed. If you look in the physiotherapy literature, you see anywhere from 0 to 80, 90% of people show improvements. And I think a lot of this is due to inconsistencies in how people apply a lot of these procedures. There's not a lot of governance of how these procedures should be applied. So our goal is to investigate whether ultrasound could be used to cause the same effect, because we can use the image guidance with focused ultrasound, align it well, monitor the therapy, and also do it completely non-invasively. So we started out in a water tank. We have a function generator amplifier. We use a Verisonics research ultrasound system for our alignment and guidance. And this is what it looks like. So, boom. So in here is, this is actually a cow tendon. Then we have our focused transducer here on the left and our imaging transducer on the right here. We have found through extensive trial and error that there are a narrow range of acoustic parameters where we can get this mild mechanical disruption. So the extreme form of histotripsy, we're literally chewing up the tissue into its submicron components, no cells left behind. Here, we're really wanting to scale it back so that we can just cause some minor fiber separation or a little bit of fraying so that we can allow these healing factors to infiltrate and cause hopefully a healing response. And so we found a few parameters that produced this mild fiber separation. The challenge was if our pulse length got too long, we started to thermally denature our tissue, which is not what we're wanting to do. So we're just trying to cause this mild mechanical disruption and see if it promotes a healing response. We have tested this mechanically. This is in collaboration with the muscle lab at Penn State. We have found that focused ultrasound does generally preserve elastic modulus of tendon better than dry needling. So our first graph here is our sham group versus elastic modulus. DN is our dry needling group, so that's one of the common therapies. And then we're comparing it to 3 different versions of focused ultrasound. And we found that blue focused ultrasound was the best. And so this was a 1 millisecond pulse repeated every 1 hertz. For a total treatment of 60 seconds. It didn't do significantly better than dry needling in stiffness or % relaxation, but elastic modulus was enough for us to be of interest. So we are just wrapping up some animal studies where we'll be able to actually measure what are our healing factors that are released and see if we're actually eliciting some sort of healing response. Meanwhile, we're parallel, we also have going trying to understand the bubble dynamics as we try to create a gel that's more similar to tendon. So tendon's very hard for us to chew up. With histotripsy, we can create bubbles, but the bubble oscillation and collapse doesn't actually chew up the tissue. Likely this is because the tendon is very elastic, so we can create the bubbles, but the oscillation and collapse isn't enough to break these elastic fibers. So we're trying some different gels. Polyacrylamide is a very common gel we use in ultrasound as a tissue mimicking phantom. We've also tried collagen gel. It was very soft, very easy to use. Fibrin gel is a little bit more similar to tendon. It's a little bit stiffer. And so we do have this effect. We're looking at the cavitation activity. So this is definitely a project we're trying to take from the basic understanding. How does anisotropy, how do elastic properties affect the bubble oscillation and collapse, all the way up to can we use this and leverage this to create a therapy for clinical use. So I'm going to wrap up there the idea of controlled soft tissue fractionation, histotripsy. We have ongoing work. We're trying to understand it for tendons. We're looking at other applications where specifically we want to try to treat these collagenase tissues. Now I'm going to change topics a little bit to talk more about imaging. So this idea of bubble-based ultrasound imaging through what's been termed the Twinkling artifact. So if you image a kidney stone with color-Doppler ultrasound, so Doppler is usually used to detect blood flow. That's what it was developed for. You get the Doppler shift, and from there you see a red or blue, depending on whether the blood's moving towards your transducer or away from your transducer. The color-Doppler ultrasound twinkling artifact was found in the early 1990s, where they imaged a kidney stone and found this rapidly changing color on this kidney stone, even though there was no motion. And so researchers have been trying to figure out since the early 90s where this artifact comes from. Is it an artifact from the machine? Is it an actual acoustic phenomenon of the wave interacting with the stone? Because hopefully you can see in this blob of grayscale ultrasound imaging, something that's showing this consistently rapidly changing color could make it very easy for people who are not experienced with ultrasound to be able to diagnose a kidney stone. So after all of those theories, we recently, in the last decade, started to get these research ultrasound systems. These research ultrasound systems allowed us to control everything that's transmitted, to received, to processed. So unlike clinical machines that are a black box of signal processing, we could use that to understand whether it's a true acoustic phenomenon. And from that, we were able to find that small stable bubbles on the kidney stone surface may cause this twinkling artifact. If you look at a kidney stone with scanning electron microscopy, so very high resolution imaging, you see that the structure is very rough with crevices and proteins and crystalline surfaces where you could imagine that bubbles could be stabilized in there. If we go back to a famous paper by Dr. Bob Apfel, we see that this idea of a bubble in a crevice for a liquid with a liquid interface, at ambient pressure, the liquid could be concave, convex, depending on which way you're looking. You increase the pressure, it changes the meniscus, but the bubble still remains in that crevice. It doesn't dissolve because surface tension stabilizes it against dissolution. If we return this again to ambient pressure, we'll just again change the curvature of the meniscus, but we still have this type of, the bubble still present in the crevice. So how could we test this? Well, we tested it by overpressure. So in 69 out of 90 kidney stones that I've tested, overpressure reduced twinkling on kidney stones, which then returned when pressure was again reduced. So the gray dotted line shows my pressure curve. The black is twinkle power. So this is really looking at the magnitude or the power of the Doppler signal. And what we see, we have this strong, consistent twinkling. We increase the pressure, the twinkling artifact immediately goes down or away. We reduce the pressure, twinkling artifact is returned at some level. So this would suggest that there are bubbles stabilized on the kidney stone. Now, of course, we could do the opposite, and instead of increasing pressure to confirm, we could also reduce pressure. So if we reduce the pressure, hypobaric pressure, we can see that it consistently increases twinkling on ex vivo kidney stones. So again, we have the dotted gray line showing our pressure curve. We have our twinkling signal, twinkle power, and it starts out at some ambient level. We reduce the pressure, twinkling increases. In this case, quite dramatically, we'd have bubbles coming out of crevices. We reduce the pressure, or increase the pressure back to ambient levels, and we see the twinkling artifacts really similar to what it was initially. So this was quite interesting, and this was a study funded by the National Aeronautics and Space Administration, NASA. And so one of the things they were curious about is if it was due to bubbles, whether the specific gas environment on the space station would affect this twinkling artifact. So carbon dioxide levels are 10 to 20 times higher on space vehicles. And astronauts are thought to be at an increased risk of kidney stone formation because they don't have gravity keeping their bones from demineralizing. So when they go up in space, their bones start to demineralize because they don't have any loading on it, they have no gravity, and that calcium from the bone goes into the kidney and may increase their risk of kidney stones. So the question is, could we use ultrasound to detect these kidney stones? And specifically through the twinkling artifact, would it be influenced by these changes in the carbon dioxide levels? So carbon dioxide is especially interesting because it does dissolve preferentially in liquids. It's 5-6 more times soluble in blood or in water than oxygen is. So we did these studies in pigs where we inserted a kidney stone, a human kidney stone, changed the gas environment, and looked at what happened to the twinkling level. We looked at 3 different levels of carbon dioxide, 0.8%, which is 20% or 20 times higher than Earth levels, 0.5%, and then 0.3%. And then we oscillated them between oxygen, which is our standard pig imaging or standard pig breathing gas for when we're doing ultrasound studies. But then we also had a group 3, that we oscillated to air instead of to oxygen and looked at what happened. And what we found was actually way more dramatic than we originally expected. So exposure to the highest level of carbon dioxide versus oxygen significantly and immediately reduced the twinkling artifact. So here we're starting, this is again our graph of twinkle power. When they were on oxygen, we had this nice solid twinkling. As soon as we went to the 0.8% carbon dioxide, twinkling artifact went away. it would try to recover, which was also interesting because the blood oxygen levels would kind of go through these fluctuations when the pigs breathed carbon dioxide. We switched them back to oxygen. It took a while, but eventually twinkling started to come back. Switched to carbon dioxide, it went away again, switched back to oxygen, and we start to see this type of increase eventually, showing that twinkling is returning. Now, to be able to compare between different animals, we looked at baseline versus middle and end of oxygen and carbon dioxide exposures. And what we found was that the amount of decrease in twinkling depended on the amount of oxygen or carbon dioxide, that the levels of carbon dioxide that were given. In other words, reduction in twinkling was the greatest for the highest level of carbon dioxide and the lowest for the lowest level of carbon dioxide. In all cases, twinkling returned, sometimes at a slightly greater level at oxygen, but in all three cases, twinkling went down, and that the reduction depended on the carbon dioxide concentrations. Now remember, we had one other group where instead of returning to oxygen, we returned the pigs to air. And so when we did that, we actually found that twinkling did not recover. So with air, it would have the same nitrogen and oxygen levels. We're just literally only switching carbon dioxide. So carbon dioxide, we see immediately starts to go down on twinkling. Air, one pig did recover, we're not quite sure why, but the other two, it continued in this negative or maybe slightly positive trend before going back. Pig 13 actually died immediately at the end of this carbon dioxide exposure. We didn't have time to switch him back to oxygen, but we're seeing this trend. We're not seeing the recovery like we saw in oxygen when we switched to air instead of oxygen. So from this, we were able to say, well, geez, not just carbon dioxide levels, but really oxygen levels also affect twinkling. And we think this is related to how the kidney metabolizes oxygen and produces urine, etc. So then this led to another question. Actually, as this was going on simultaneously, the question has always been asked, are there these microbubbles on kidney stones in humans? And this question comes because all of our ex vivo stones, well, yes, we would put them back in water, degas them, try to remove any bubbles that we may have artificially added. We never knew whether there would actually be bubbles in the human body. And so imaged 8 human subjects in a hyperbaric chamber. And this was a big hyperbaric chamber, about the diameter of a 737 aircraft. So we could put the sonographer in there, put the subject in there, and image them. Because of the risk of fire, we had our ultrasound system outside. The sonographer got to view the ultrasound image through a small port window in the wall, and from there be able to track what's happening. Now, this is the pressure profile we got approved for. So we'd start at 1, this is atmospheres absolute. So this is just normal atmospheric pressure. We would ramp up quickly to 3 atmospheres absolute, have a pause so we could see what's happening to twinkling, then ramp up to 4 atmospheres absolute. For those of you who might dive, that's the same as diving to 99 feet, after which we would start to reduce the pressure. Now, because we put healthy people in, we wanted healthy people out. So we had this pause at 1.6 atmospheres absolute, where they breathed oxygen to reduce any risk of decompression sickness. And then finally look at what happens post-study or at the end for the twinkling level. Now, for those of you who don't really appreciate how much pressure we're adding, we took a football into the chamber once, and before pressure was added, it was fully inflated. At 4 atmospheres absolute, it went down to about half of its thickness or half of its inflation level. So similarly to what we found in pigs, we found these twinkling changes over time and pressures. So here in green, we have our pressure profile in atmospheres absolute, and blue is our twinkling power. So what we saw in our first subject, we do get this decrease. Again, there's some oscillation, but generally this decrease in twinkling. But even more interesting than the decrease was we found that twinkling actually increased when they were breathing oxygen at 1.6 atmospheres. This happened also in subject 3, where we sort of see a decrease, but even more interesting, we see this increase when they breathe oxygen. If I aggregate the data from all 7 subjects, one subject we had to exclude because the data was corrupt. But the other 7 subjects, we do find that this reduction in twinkling is actually significant. So I normalize it to the baseline level. Blue is our pressure. Twinkling is significantly reduced with a p 0.4 for both 3 and 4 atmospheres absolute. we didn't quite get a statistical significant increase when they breathed oxygen here, likely because some of the subjects had a huge influence on oxygen, some of them had no influence on oxygen. So clearly there's some other roles that we have to investigate. And then finally, Twinkling did return to its approximate pressure change levels. So taking this work even further, we're trying to understand here at Penn State what specific stone features cause the twinkling artifact. I've shown you all the really cool stuff, but we're still facing this hurdle that twinkling only appears in about 60% of kidney stones. And we're trying to understand whether those 60%, whether it means something, whether it can indicate some sort of composition. What does it mean that only 60% of stones show this twinkling artifact? So one of the first projects here we tackled at Penn State was trying to understand where does twinkling occur versus what structures cause twinkling if we try to use other technologies to look at the surface architecture. So here we see we scanned across the stone at 3 different frequencies, a couple different stones, and we see some isolated areas of twinkling or high Doppler power. That's the red area showing that we get twinkling there. And then we try to compare that. So how much of the stone volume shows twinkling? And so here we're just saying that, well, some stones show a lot of twinkling and some stones, as we'd predict, don't show very much twinkling. Generally, the lower frequencies, 2.5 and 5 megahertz, showed a lot more twinkling than the 18.5 megahertz. And we then looked at it with scanning electron microscopy, but not just normal scanning electron microscopy. We used environmental, where they changed the humidity so we could really look at where these crevices are, because when the bubbles or when the water coalesces indicates that that's where the first place bubbles would be likely to form. So we found that each stone had very different surface structures. So a very smooth calcium oxalate dihydrate stone was very different from a struvite stone versus a jagged calcium oxalate dihydrate. All of them were very unique, even for 2 stones that were supposedly the same surface or same chemistry. And we compare that to the amount of stone that twinkles. Surprisingly, this one that doesn't look like it has too many jagged surfaces had more twinkling than the struvite stone, where we see these very rough jagged surfaces. So trying to understand what that means, we said, well, maybe twinkling isn't just caused by bubbles on the surface, but maybe it's also caused by bubbles inside the stone. So we looked then at these internal crevices and micro tracks. So using a fancy micro CT unit that had enough power that we could keep these stones submerged in water and image them both in ambient and hypobaric conditions, where hypobaric, we'd expect those bubbles to get larger. So we see these dark regions. When we apply hypobaric, if those are actually bubbles, dark regions, we would expect them to expand. And so in this case, we didn't see much difference in those dark hypobaric regions. And here's just a nice animation showing what happens. The top is ambient pressure, bottom's hypobaric, not seeing significant differences between these dark voids. But in this smooth or struvite stone that had a little bit more twinkling, we find that these dark cracks and crevices, when we reduce the pressure, expanded significantly. So this would suggest that there are bubbles trapped inside these stones that may be contributing to the twinkling artifact, because the surface didn't explain it all on its own. And finally, here's one other stone. This stone also had cracks and crevices throughout, but we didn't see any change when we pulled the vacuum. So these crevices, while they look like they could also be full of air, actually appeared to be mostly liquid because there was no change when we changed the pressure. So this suggests, it doesn't prove yet, but suggests that internal characteristics may also contribute to twinkling and may explain why some kidney stones didn't show the effect of pressure like we'd expect for surface structures, and may also help lead us to why some stones do not show this twinkling artifact. So what we've shown so far is that results continue to support this idea that bubbles are at least on some stones and cause a twinkling artifact. Now, these bubbles may also be inside the stones in these micro cracks and crevices, but they're definitely on the surface of the stones. Bubbles can be directly observed if we enlarge them. We can use pressure changes to enhance or reduce twinkling. And we can see that these different gas compositions also enhance or reduce twinkling. And that's something we're looking at following up on, is for places that don't have access to CT X-ray, could we just have somebody breathe oxygen to enhance twinkling to be able to better diagnose kidney stones? Nevertheless, that human subject study was the first evidence that bubbles could exist as bubbles, stable bubbles that aren't just appearing and disappearing in the body. And so that was a really exciting conclusion from that. Now we're taking this work beyond our initial start on kidney stones. There are other places that minerals form in the body. Cholesterol crystals can form in these atherosclerotic plaques, or these plaques that form in arteries, and may indicate that the plaque is more prone to rupturing the vessel and causing death, or at least extreme consequences. We get uric acid crystals that form in the joints in gout. We get calcium phosphate crystals that can form in something called heterotopic ossification, where bone is deposited in places where bone really shouldn't exist. All of these different crystals that we've grown in the lab do show a twinkling artifact, with cholesterol showing the most twinkling, calcium phosphate and uric acid showing a little bit less twinkling. But in general, we're finding that this twinkling artifact is very sensitive to the presence of crystals. Now, specifically heterotopic ossification, we are looking at whether and how early we can detect this bone being deposited. So we have some evidence that we can have these growth cells that can be caused to start depositing minerals very early. And we're looking at these with the twinkling artifact. So here's a small layer of cells on a scaffold, and we start to see this twinkling artifact that we're working to link to this green area, which is showing the minerals deposited. The blues are the cells. So how sensitive are we to be able to detect these minerals is what we're looking at establishing, because if we can diagnose something like heterotopic ossification early, we could hopefully avoid some of the extreme consequences for some of these diseases. We're also looking at twinkling to detect phase change or microbubble contrast agents. So with the Medina lab, we've shown that these phase change contrast agents, literally they're the droplets filled with these perfluorocarbon gases, where they're liquid below body temperature, and they have a phase change when they go up to body temperature, 37 degrees Celsius or more. But basically, you can unload drugs and other things on in these droplets. The droplets are small enough they can get out of the bloodstream into tumors, and from there being able to deliver drugs and cause inertial cavitation to try to break up tumors and etc. So we did see that the twinkling artifact was also useful to detect that phase change and be able to hopefully quantify when they undergo inertial cavitation. And we've worked with their group and another group at Penn State, the Manning lab, to show that we could use Doppler ultrasound with those phase change nanodroplets to be able to evaluate clot boundaries in deep vein thrombosis. So here we have a case with no nanoparticles. We see we get this circulating behavior because there's a clot that's obstructing this vessel, where once we have these nanoparticles, we get a little bit of a clot, and you get this twinkling artifact because we're activating those nanodroplets right at the clot boundary. But this actually, when we tried this, it also worked to inhibit clot from forming. So it had two roles, where it both outlined the clot better, but also prevented the clot from forming. And using our research ultrasound system, we can recreate these flow profiles to try to understand better the kinetics of how a clot forms, and how you could use something like a phase change contrast agent to both inhibit clot formation, but also deliver the drug more efficiently. Finally, we have a project we're working on the basic science side to try to understand what is the distribution of bubble nuclei for acoustic cavitation in tissues. So more and more people are looking at these cavitation-based therapies for many different applications of ultrasound, but we don't really know where a bubble forms. Are they going to form inside the cells, outside the cells? Does that matter as far as the therapeutic effect or the risk of off-target injury? So Ferdusi, one of my newer students, is starting with these gels to try to understand how consistent is bubble nucleation site in gels, and does it affect it if we change the stiffness, change the peak negative pressure? And so we are finding that, of course, as we'd predict, that where cavitation occurs becomes more consistent as we get higher peak negative pressures to literally pull apart the the gel or tissue samples. And we're doing this as we work into some cell studies where we'll hit those with ultrasound and say, are the bubbles more likely to form inside the cells, outside the cells? Does it depend on the type of cells, et cetera, to really understand what's happening at the fundamental level of bubbles as they form in tissue. So hopefully I gave you a nice overview talking about two specific areas, controlled soft tissue fractionation and using that for cancerous tumors, for physiotherapy, for tendon injuries, but then also this other project talking about bubble-based ultrasound imaging and how we can apply it to different areas, potentially use it to treat the areas, and making basically a better image for people to be able to follow and use, even if you're non-expert at using ultrasound imaging. So all of these technologies, I think, could be faced on, of course, on Earth, different areas of Earth, developing countries and space, et cetera, and of course, developed countries. So all of these same physiologic challenges occur on Earth and in space. And ultrasound is one of the few technologies that has both diagnostic and therapeutic capabilities for both Earth-based applications and for spaceflight. And so the proposed research, we hope, has the potential for clinical implementation. We are working with the clinicians down at Hershey Medical Center and are looking at how we can transition these into spaceflight and other areas where there are low resources for medicine. So this work has been supported by National Space Biomedical Research Institute, National Institute of Health, National Science Foundation, and the Congressionally Directed Medical Research Program. I'd like to thank you for your attention. This is an old picture of my current lab and an old picture of my old lab. A lot of people go into making these work, but if you have would like more information on what my lab does, we have a website, sites.psu.edu/BASiL. Thank you for your attention.
Well, I would like to thank very much Dr. Simon for this fantastic presentation and research, so interesting. We'll see if we have any questions in the Q&A. We do not. I do have a question, but I was waiting until someone else showed up. But my question is more on the fundamentals of the physical process that occurs when a bubble collapses or collapses. So what happens? There is a release of energy. What is the physical nature of the process?
Yeah, so as soon as you have a bubble that's excited by an ultrasound wave, if your ultrasound wave is relatively low amplitude, that bubble can just oscillate in and out continuously and not cause any big effect. It can cause some shearing at cell membranes and break up some cells, but it's not going to cause any big damage. But as you increase the amplitude, eventually you get where the bubble tries to expand in response to the negative pressure, and it has to expand so fast or so big, you literally get a vacuum inside the bubble. And once you do that, it's going to collapse very violently. And that violent collapse can either happen through these jetting, where you get the jetting towards a surface, you can cause pitting. This is what happens in a lot of ship propellers, where they get this cavitation damage because of that spinning and they get that negative pressure. You can also get just the violent collapse. So it's just going to hit down really hard and rebound. And just that energy release as it hits down and rebounds also can be used to break up tissues, cause damage to ship propellers, et cetera. It's a very violent activity.
Yeah, it's a mechanical effect, totally mechanical.
Yeah. Completely mechanical.
Yeah. No chemistry attached to it. No release of liquids or nothing chemical. Just mechanical.
It's just mechanical, though a lot of people are developing these microbubble contrast agents where you can put drugs and other things in so that when that bubble collapses, you are releasing some sort of drug into a local environment. So that's an active area of research in a lot of biomedical acoustics, is how to locally release these toxic drugs into cancer cells, basically.
Very interesting, really interesting. See if any of our participants has any other questions. So, well, I don't see any other questions. Well, there seems to be no further questions. So I would like to thank very much Dr. Simon for presenting this cutting edge research, and always in the committee of our chapter, we're discussing how to reach out to other areas of acoustics that are not architectural acoustics, which is our we have an abundance of architectural acoustics in our members and also in our presentations, and this is reaching out to other boundaries of acoustics that are also important and people should be aware of, I think. So
fantastic. Thanks so much. And if anybody has any interest in collaborating or whatever, you can find my email at this site and we can figure out a way to collaborate.
Yeah, sure. Well, without further ado, I will stop the transmission. Thank you once again, Dr. Simon, and in the name of the Spanish-speaking acousticians, I thank you very much for your presentation.
Thank you.